Okay, so I um, studied the biocatalytic approach of synthesizing islatravir. So islatravir is currently still in its investigational stage, but it's a drug that's a solution for HIV infection, and it's considered, these are like a bunch of different words that bind it together. It's considered an NRTI, which stands for nucleoside reverse transcriptase inhibitor, but it also incorporates translocation, just a bunch of different processes of like RNA and DNA synthesis and transcription and everything. So to understand how this works, it's helpful to understand how HIV works. So the genome in HIV is RNA instead of DNA. So what happens is um, reverse transcriptase catalyzes reverse transcription, which is going to turn RNA into DNA. Um, and then this is how like HIV replicates and like gets into our systems and everything. So islatravir prevents this from happening, and uh, hence like reverse transcriptase inhibitor. So this is the retro like salvage pathway. So it's kind of like a planning tool, but it's not necessarily like a reaction scheme. So what happens here is these arrows are like important in how they're set up. So this indicates a retro reaction. So it's basically saying like, for example, we can make um, 3 from 4 using ppm. So it's kind of like thinking backwards than what you would expect. So it's not exactly like a laid out reaction. It's just more so a planning tool. So it's helpful to know like what each of these enzymes do in order to understand how this pathway works. So PNP is purine nucleoside phosphorylase. So it's just going to displace the nucleobase and replace it with a phosphate, deoxyribose 1 phosphate. So we can see that here. It's adding just like the phosphate. And then PPM is going to transfer the phosphate to the 5 position, and that resulting substrate is going to become, or that resulting sugar is going to become the substrate for DERA. So they're all kind of like backwards here. But here it's saying like this is going to be the substrate now for DERA. I believe I've interpreted that like that. And then DERA performs like a retroaldol reaction into glyceraldehyde 3-phosphate and acetaldehyde. So the mechanism for the DERA reaction looks like this. So some amino acid is going to take a hydrogen from the methyl group on this acetaldehyde, and just going to perform an aldol reaction here, binding these electrons to this carbonyl. And then an intermolecular reaction is going to occur to make this a ring. So we see this here, and this is what we see here. So that's what the DERA does to form that ring. So now this is another reaction that they focused on. This was like a further process that they wanted to touch on to like, you know, complete this. So this is our starting material here is 6, and they really wanted 5. But they didn't know which order to do, if they were going to do the phosphate transferase first or the oxidoreductase first. So I believe I don't know if there was full reasoning behind this, but I think they took this pathway, the oxidase first and then the phosphatase second. That's the one that they used mainly. And it was important in this that they get the correct stereochemistry for this hydroxyl group and alkyne group. Yes, they didn't really know which one was best, but this is the pathway that they did more often in the research, was 6 to 7 to 5. So this is the table that highlights a lot of what they were looking for in their process and what the results were. So this rounds of evolution, if you remember one of my previous presentations, there was a whole chart, and it was like each line, there was different mutations. So each line here indicates one round of evolution. So a round might have anywhere from like 1 to 3 or 4 mutations. So this first one had 12 rounds, which is like a lot. And what they wanted to do here was basically make the protein better using E. coli instead of this Fusarium graminearum, and GoAce was just like the oxidase enzyme that they used. And the loading percent changed, but it didn't change very much. I think it changed like 5-fold. But the loading percent is basically how much enzyme is needed to make the substrate do what you want it to do. So they also have here evolution focus, so what they focused on, what their goal was for training the enzyme to do. So along with the activity of, making it work in E. coli, they focused on stereoselectivity. So they really wanted, if we look back in this, like I said, they focused on this having the proper stereochemistry here. So that was a big goal of training this enzyme through like those 12 rounds of mutations to do that. And they were very successful in it. This PAN-K, they focused on the activity of it. So you'll see like that the loading percent here hasn't changed, but what did is this percent, so this is percent yield. So before they had, less than 1%, and then after they've done all these 3 rounds of evolution, the 3 rounds of mutations, they have greater than 95% yield, which was a huge, great increase. Here for DERA, they focused on acetaldehyde tolerance. So aldehydes are really damaging to proteins, so they were worried that having a lot of this acetaldehyde was going to ruin the protein, so they wanted to train the enzyme to be able to handle it, and they did it perfectly. And the loading percent, it looks like it hasn't changed a lot, but it really changed 25-fold, which was really impressive. And that was basically the only thing that changed, and it only took 2 rounds of evolution, which I thought was pretty cool. And then the ppm was mainly just percent yield. You see here, it increased a little bit, and then the same for PNP. So those 2 I didn't find as impressive, but they're still very important in the reaction. And then this was their final scheme. So basically these are all their evolved enzymes that they trained to do certain things, and these are the auxiliary enzymes. So what those do is they basically prevent the steps from being reversible. So for example, if you look at SP here, it's gonna remove this phosphate and just have it attached to glucose, into some form of glucose, so that it can't really, go backwards in this case, and but it doesn't affect the islatravir. So the islatravir is made, it's safe, it's happy. They're just gonna remove the phosphate, again, just to clarify that it won't go backwards. And then here they have said somewhere the immobilized version of an enzyme. So that's just here we see the GoAce. This purple little dot represents a bead. So it's it can be reusable and makes it very efficient and very green. So they were able to work up the reaction, very easy process. These evolved enzymes. And yeah, so overall, they were able to, train enzymes to do what they wanted them to do, and they were pretty generic enzymes, but they just wanted a pathway first using the retro laid out plan. But they were able to train them to do what they wanted, and they were very successful. And they made a complicated molecule, this is islatravir, through a very simplified process. So it's very revolutionary, I thought, and just very efficient overall. But I think that's kind of all I had to say for now.
So does anybody have any questions? It's a little, Emily had a big task, because the way that they sort of displayed it in the first figure was using these retrosynthetic arrows. Are you all familiar with what that means? What those are? Emily said it, but it does, I think it does take a minute to sort of get used to it. That the arrow, well, first of all, as you might know, there's a bunch of different specific arrows that people use for different things in organic chemistry. There's like a regular arrow just means like a reaction arrow. Resonance arrows are special because they have barbs on both ends. This arrow actually, it's like two lines and then a barb on one end. That is a special kind of arrow that means that's a retrosynthetic arrow. So what that really means is that they are, the product is on the left and they're disconnecting it into things that they think they can put together in a forward sense, you know? So if you want to, the easiest way to understand this is that their planned route actually begins at the right? Compound 5 and the acetaldehyde. And they think they can put those together using the DERA enzyme to make compound 4. And then they think that they can treat compound 4 with an enzyme to make compound 3. And that they think compound 3 they can make into islatravir by combining it with compound 2. So it's just a plan, but that's their sort of retrosynthetic plan because they have enzymes that have all of those sort of general activities that they are confident that they can train to get like improvements. Do you know what I mean? So I don't know. I think Emily, it might not be a bad idea to just talk like in a forward sense, like from right to left. Just maybe name the enzymes and maybe just say one more time like what they are doing, I guess.
Yeah.
In a forward sense, because it's just less confusing than the backward way. Do you know what I'm saying?
Mhm. Yeah, so DERA does the retroaldol cleavage. So that's the mechanism that I drew out. So it's going to make this glyceride 3-phos. It's going to use the glyceraldehyde 3-phosphate and the acetaldehyde. So DERA is undergoing this mechanism to make this compound.
And there's another sort of limitation here because like they called it like a retroaldolase, which is just the last two letters in the abbreviation. But of course they needed to do not a reverse aldol reaction, but a forward aldol reaction. So that's where they have to kind of like, that's a big problem that they have to sort of fix is like the equilibrium position of what the reaction that the enzyme catalyzes is.
Yeah.
But okay. And
And
then
like allowing high tolerance or high concentrations of this acetaldehyde so that it doesn't like disrupt the protein in the process either.
correct, right? So DERA needs like a lot of work, right? It has to be able to tolerate that acetaldehyde, and then it has to make 4 pretty efficiently from 5. So, and I think we might have to revisit this later because that pan-A enzyme, Emily, I think is trick that they used to get the reaction to go in the 5 to 4 direction as opposed to the 4 to 5 direction.
Okay.
Because if you look at the other scheme, they actually don't start with acetaldehyde. Can you just flip down real quick? There. Yeah, do you see how oh, I'm sorry, I made a mistake. They totally do. So I don't quite understand it yet. Okay, can you go back up? Because what I was gonna say is totally wrong. Thanks. Yeah, and then what's the 4 to 5 reaction, just so people's eyes can see it.
So now ppm, that's the phosphopentomutase. So that's going to transfer the phosphate group to the 5 position. So that's putting the phosphate, like that's just around, it's going to put it here. So this is like the 5 position on the molecule. Right.
Yeah, so actually, again, it's confusing because, again, they're describing the reverse reaction than what they want. So the 5 position is where it is in 4, and they want to move it to the 1 position.
Yeah. Okay, yeah. Okay,
That's
so
the problem.
I also might have wrote that down wrong. So, okay, yeah, so the phosphate was transferred like out of this position and a hydroxyl replaced it. That's what the ppm does
Yeah,
in
it
like the
moved it.
forward reaction.
Yeah, it moved it to 1.
Right here.
See how the carbons are numbered in the 4 structure?
Yeah.
So it moved from 5 to 1, yeah.
So these will be replaced using the ppm. And then the PNP, that's gonna, in the reverse reaction, displace the nucleobase with the phosphate deoxyribose 1 phosphate.
Yeah. And again, unfortunately, they're describing the reverse reaction than what they want. You know what I mean? Because they want the nucleobase to displace the phosphate, not vice versa, have a phosphate displace the nucleobase. Do you see what I'm saying? I think you froze up. Emily. Oh, maybe I froze up. Am I frozen up? Who's frozen? Can you hear Emily? Oh, I think Emily had internet problems.
Yeah, Emily's frozen.
But it's unnecessarily confusing because the names of actually every single enzyme in here is named for the reverse reaction than what they're trying to get it to do in their sequence. So it makes it confusing when you're calling something like a retroaldolase, but you're trying to make it do like a forward aldol reaction and so on. There she is. Hey, Emily.
Hey, sorry, I think I lost the internet. I'm sorry, where did you last hear?
There's just the last reaction to make islatravir.
Yeah. Okay, so just to like fully reiterate, these two combine using the DERA, which is going to make this, and then the phosphate and the hydroxyl are basically going to switch using the ppm, and then the phosphate here is going to be replaced on the opposite face with this nucleobase using the PNP, and then that's going to make islatravir with like phosphate kind of just floating around.
Yeah. And this is sort of, I think the strategy is sort of like inspired by like a natural pathway that bacteria use to take nucleosides and basically degrade them into like simple starting materials that they can use for other things. So they're trying to run that the breakdown pathway in the reverse to make one, right? So that's probably why all the enzymes have the opposite name of what they want them to do. You know what I mean? So it was like, I don't know. I mean, when I read this paper, I sort of thought about like the committee meeting where these scientists were trying to, convince their bosses to let like 15 people work on this for 2 or 3 years. And then they were like, so you want to make islatravir, you want to engineer a reverse pathway, and then you need to train these enzymes, and then you need to get them to do it with unnatural molecules with this weird alkyne, and you don't want to do any chemical reactions, like, right? You want it to be all enzymes. And then the scientists were like, yeah, that's exactly what we're saying. And then they said, okay, like have at it. And then they did it. You know what I mean? Because I want you to notice that there's no non-enzymatic step here. Nobody's freaking out, Emily. I don't know what the problem is. You know what I mean?
Yeah. Super green, super cool,
Yeah.
super.
Okay, so maybe just you and I can freak out about this. That's okay. I mean,
This is amazing, guys.
yeah. But the one thing that I think, the sort of the part of it that I think is going to require a little bit of, I think maybe you should do on like another day, is to talk about these auxiliary enzymes that are not in the pathway, which are, there are a bunch of them. The catalase, the horseradish peroxidase, the pan-K, which they gave a color to. Do you see what I'm saying up there? For between, to make 5 from 7. Yeah, that's actually not one of the ones in their scheme up top. It's like a different one. And then also at the end, the sucrose phosphorylase. Because that, I think all of those things, in addition to the engineering that you talked about of the core enzymes, the DRA and the phosphoMutase and the purine nucleoside phosphorylase,
Hmm.
I think those things are the things that turned it from an idea into like a reality. You know what I mean?
Yeah.
That in order to get things to move in the right direction, oftentimes I think using Le Chatelier's principle, to, like you said, with the phosphate, to basically get rid of the phosphate so it can't reverse. Yeah. All right. Questions? Can I show you something real quick? Islatravir looks a lot like remdesivir. Do you know what remdesivir is?
That sounds familiar. I think I've talked about that, but I
It's
forget.
a potential drug against COVID.
Oh, yeah.
No, it's not even potential anymore. It's out
Are they
there.
actually studying it?
I just want to show you, this is, I'll share screen in a sec here. I want to show you how similar the structure of remdesivir is to islatravir. Hold on here. So, okay. So can you see my screen? So this is remdesivir. Actually, let me go back. I'll go to remdesivir. All right. So this is remdesivir, but all of this junk over here just comes off inside of your cells. It's like a prodrug. So all that stuff has to get cut off to make the active drug, which is this GS. Here it actually says it is a prodrug that is converted into the body into GS 441542, which is this compound. And I want you to notice that this is almost islatravir, right? There's a couple differences. Islatravir is a deoxynucleoside, so it lacks a hydroxyl group there. The position of the nitrogens in this base is unnatural. They're at alternate positions, and actually, typically, it's a nitrogen that's actually added to the ribose. So this is called a carbonucleoside. And then, like islatravir, which has an alkyne here, GS 441524 has also an sp hybridized group, but it's a nitrile instead of an alkyne. And it is also, let's see what I believe it is also. I'm trying to find its mechanism of action. I believe it is also a reverse transcriptase inhibitor. So it does the same thing. If I go back to remdesivir, it probably will tell me. Mechanism of action. Oh, no, it's different. I'm sorry. It's an inhibitor of RNA-dependent RNA polymerase, which is not actually super dissimilar from reverse transcriptase. So anyway, it's similar.